The bright field microscope is the most common microscope used in laboratories. In its simplest form, it's just one magnifying lens. This is basically a magnifying glass, and this is the type of microscope Leeuwenhoek first made. Today we use compound bright field microscopes or compound microscopes, which use a series of magnifying lenses. Typically there are two, the ocular or the eyepiece, and then the objective. Bright field microscopes have a condenser in them. This condenser helps make all of the light that gets to the condenser travel in a straight line through the specimen. Otherwise, the light would be somewhat scattered. To determine the total magnification of a bright field microscope, you simply multiply together the magnifying power of each lens, in this case the ocular times the objective. Oculars typically have a magnifying power of 10 times. Objectives will vary. 4, 42, 100 are very common objective magnifying powers. One of the problems with the objectives is when we increase the magnification, we actually make the diameter of the lens smaller. And this means that we can lose light. The light starts to dim as you go to higher magnifying powers. At the highest magnifying power we typically have with a bright field microscope, 100 times on the objective, we tend to lose a lot of light. And so we have to use oil on the slide to help with keeping the light directed to your eye. This is because light does something called refraction. When you have light passing through the glass slide and back into air, the refraction of light or the bending of light means that some of that light now does not hit or go through the objective. So what we want to do is put oil on the slide. Oil has the same refractive index as glass, so as light travels through the glass and into the oil, it doesn't bend any further, and this keeps more light traveling through the objective and to your eye. Bright field scopes are very good for examining stained specimens. However, they can be used to look at unstained living specimens as well. The dark field microscope has a special stop in the condenser. What this does is it directs the light directly onto the specimen. It sort of puts the specimen in a spotlight. The field appears dark, but the object on the field is lit up. Magicians use this technique a lot when they spotlight what's going on with the magician, but all the back of the stage is dark. Lots of stuff can be going on in that darkness that you don't know about. Dark field microscopes are particularly good for cells that don't stain well. Either they don't go through the heat fixation process well or they don't take dyes well. Spirochetes are a group of bacteria that typically are best demonstrated using the dark field microscope. Phase microscopes use the fact that when light passes through a specimen, the light that passes through the specimen slows down just a little bit. This means that light is no longer in phase. Those peaks and troughs of the wavelength of the light are a little bit out of sync, or we say they're out of phase. Your eye can actually detect this. If we add a special lens to the microscope that enhances this phase difference, we come up with a phase contrast microscope. This creates a contrast between the specimen and the background. There are two types of phase microscopes. One is the phase contrast microscope, which gives you a pretty sharp 2D image, and the other one is a differential interference microscope, sometimes called a Nomarski microscope, that gives you a 3D appearance. Now here you see the same specimen using different microscopes. This is your bright field, and you can see a little bit of detail, but not much. Then here's dark field, and a lot of the pieces now are lit up, not because of anything we've done to them other than how we've altered the light hitting them. Here's your phase contrast, nice sharp images. See how we have more contrast than we had here? And then here's the Nomarski, that 3D kind of image. Fluorescence microscopes use ultraviolet light as their light source. Ultraviolet light is a shorter wavelength than visible light, so it increases the resolution quite a bit, but we can't see it with our eye. Therefore, we have to use something, some material that will absorb the shorter ultraviolet wavelength and then re-emit light at a longer visible wavelength. These are referred to as fluorochromes. Now, some cells do this naturally, and some cell structures do this naturally. Chlorophyll, for example, naturally fluoresces. If you look at it under a fluorescent microscope, you'll see chlorophyll light up beautifully. But for other things, we can use special fluorescent dyes, and we can even make these dyes specific for an organism or a structure. This is a process called immunofluorescence because we're going to bind the dye to antibodies, that's the immuno part, that are specific for the organism. The dye will only be taken up then by that particular organism, and that is how we will see it. So here we see antibodies and our fluorescent dye, and we bind them together. And if these antibodies are to a particular organism, they will attach to that organism. The dye will be attached to the organism as well. And when we look at them under a fluorescent microscope, the organism lights up. Mycobacterium tuberculosis is an organism that uses fluorescent staining to demonstrate it. Here, it's very difficult to see the cells in this specimen. But when we stain it with a fluorescent dye, only the cells that picked up the dye show up. And because we were using an antibody to Mycobacterium tuberculosis, only the Mycobacterium tuberculosis organism picked up the dye. Confocal microscopes are a special kind of fluorescent microscope. They use fluorescent dyes or antibodies as their stains, but instead of just plain ultraviolet light, they use ultraviolet lasers, so they illuminate only thin layers of the material. They take pictures of each layer, and then they can put the pictures together and construct a 3D image. Electron microscopes generally magnify 10,000 to 100,000 times. At this magnification, you can see viruses as well as cellular ultrastructure. Very special ultra-thin preparations of materials are required for electron microscopy, so the preparation of these materials is very time-consuming. Electrons travel in a vacuum tube, and since they're all negatively charged, they tend to bounce off of each other. So we have to focus the electrons with electromagnets so that the electrons all travel in straight lines. So an electron microscope looks nothing like any kind of microscope you've ever seen before. Transmission electron microscopes give you a 2D image of something, and a scanning electron microscope gives you more of a 3D image. There are some new electron microscopes called probe microscopes. The magnification here is greater than 100 million times. This is tremendous magnification. There are two types of probe microscopes. One is the scanning tunneling microscope, and this has a metallic probe that has been sharpened to 1 atom. The tip of that probe is 1 atom. This will travel close to the surface of whatever you're looking at, and it will measure the flow of electrons between the probe and the specimen surface. This is a little bit like sonar, where we determine how long it took the electron to travel between the two, and we can kind of map the surface of something. Now, the specimen has to be electronically conductive for this to work. The atomic force microscopes have the probe travel directly on the surface of the material they're examining. This is sort of like someone reading Braille. As a result, the specimen does not have to conduct electrons, and it can be used to look at living material because a vacuum is not required. Staining provides contrast for us. It helps us see things better when we magnify them. And staining is simply coloring cells to help make them more visible. When we prepare a slide, it's called a smear. And to make a smear, you simply put a thin layer of cells on the slide, let it air dry, and then you fix the material to the slide. And you can do this with either heat or chemicals. Drying is called desiccation, and the desiccation and fixation process helps kill the microbes. Not all microorganisms will be killed by air drying them. It helps attach them to the slide so they don't wash off during the staining process, and it helps preserve the shape and size of the cells so you get a true picture of what you're looking at. There are several different kinds of stains. Simple stains are just single dyes. You soak the cells for 1 minute, and the cell wall is colored. It's a lot like coloring Easter eggs. What you get is a nice picture of the cell shape or the cell morphology. The cells will all be the same color, but you'll get to see their unique shape. Differential stains use two or more dyes, and they allow for some sort of categorization of the cells. Something about their color tells you something additional about them other than just their shape. The common differential stains in the clinical microbiology lab are the Gram stain, the acid-fast stain, and the Endospore stain. Now, histological stains, stains of tissues, can be used to identify microbial elements in tissues. These are typically not done in the microbiology lab, but a couple of common ones are the Gamorrean methenamine silver and the hematoxylin and eosin stain. The Gram stain is the most common of all the differential stains. You're going to start with crystal violet. Crystal violet is your primary stain. You are simply going to put that on the smear and let it sit for a minute. Now, you wash it off at this point. It could be a simple stain. All of the cells are going to be purple in color. This simply adheres to the cell wall. Next, you add Gram's iodine. We say Gram's iodine is the mordant. Gram's iodine binds to the crystal violet and forms a large insoluble molecule that's very difficult to wash out of the cell. Again, if you stopped at this stage, you would still see all purple cells. The decolorizing agent is usually acetone or acetone alcohol. This is going to dissolve any lipids that may be in the cell wall. All bacterial cells have peptidoglycan in their cell wall, but they don't have the same concentration of peptidoglycan. Gram-negative cells also have a fairly high content of lipopolysaccharide. This lipopolysaccharide acts as the endotoxin for these cells. The endotoxin is what makes people generally feel yucky if they have a gram-negative infection. They feel bad, they have a fever, those kinds of things are the result of the endotoxin. But this lipopolysaccharide is soluble in acetone and alcohol. Gram-positive cell walls have a very large peptidoglycan content and virtually no lipopolysaccharide. When we flood the slide with this decolorizing agent, the gram-negative cell wall has large holes washed in it as a result of that lipopolysaccharide dissolving, and these holes are large enough to let the dye complex wash out. If we were to stop the staining process at this point and look at the slide, we would see purple cells that were the gram-positive cells, but the gram-negative cells would be invisible. There wouldn't be any color to them. So we finish off with safranin, which is a counter stain. Safranin will be picked up by those colorless cells, giving them a pink color. The gram-positive cells are already stained a much darker blue-purple color, so the safranin will not alter their color at all. When you look at a gram stain, the purple or blue cells are the gram-positive cells, and the pink or red cells are the gram-negative cells. So we have two different kinds of organisms. We have gram-positive and we have gram-negative. In addition, we can still see them, so we still see their cellular morphology. All of these are rod-shaped, so they're bacilli, but we get another piece of information. And these bacteria are a little different because of the composition of their cell wall. The acid-fast stain is particularly helpful when working with the genus Mycobacterium and the genus Nocardia. These are still bacterial cells. They still have peptidoglycan in their cell wall, but they have a large quantity of this waxy lipid in their cell wall. And this waxy lipid keeps them from dehydrating. It allows them to survive rather extreme circumstances, so this is part of their protective defense mechanism. But wax is very difficult to stain. So the classic acid-fast stain is called the Ziehl-Neelsen stain. You use a primary stain called carbylfuchsin, but you have to steam this on the slide. So you put the dye on and then you use a Bunsen burner until you see steam coming off of it. And you have to steam it for about 5 minutes. The idea is the wax melts a little bit and the stain seeps into it, so the wax is going to retain the stain. Because you've taken some pretty drastic measures to get this carbylfuchsin into the cell wall, you need to decolorize with a weak acid. The wax will still retain the stain, but all other components of the cell will lose the stain. Then you counterstain with methylene blue. The idea is acid-fast cells will be pink, non-acid-fast cells will be blue. Since all of your acid-fast cells are rod-shaped or bacilli, we talk about AFB, acid-fast bacilli. And here you can see the pink cells. And then the non-acid-fast cells are the blue ones. Now, while this looks like this is easy to see, in real life it's not. Acid-fast bacilli are very challenging to see in a clinical specimen. So more commonly, instead of using the Ziehl-Neelsen method, we use a fluorochrome stain, an auramine-rhodamine stain, that's much more specific for the organism and easier to see. Some organisms, particularly the genus Bacillus and the genus Clostridium, produce endospores, which are very dormant but highly resistant structures that allow them to survive long periods of time in very harsh conditions. These endospores are very difficult to stain. Now, if you stain these cells and they have endospores and you stain them with a gram stain, you may see clear spaces in the cell. But if you want to demonstrate that those clear spaces are really endospores, you use the Schaefer-Fullerton stain. Again, this is one of those stains you have to steam on because you've got to crack the wall of those endospores to get the stain in, and the stain you use is malachite green. Here you can simply decolorize with water and then counterstain with safranin. The endospores will stain green, but the vegetative cells will be pink or red. Histological stains, as I said, are not typically done in the clinical laboratory. They are used on tissues. The Gamori methenamine silver stain will particularly help show fungal elements in tissues. And the hematoxylin eosin stain is a pretty much general purpose histology stain, but if there is any kind of microorganism, it will show up using the hematoxylin eosin stain. Special stains are used on structures that may be challenging to reveal on microorganisms. The most common special stains are the capsular stains, the flagellar stains, and the fluorescent stains. Now, the capsule stain is what we call a negative stain. A negative stain stains the background but leaves the organism unstained. All of the other stains we have talked about have been positive stains. Positive stains stain the organism and leave the background unstained. Now we can use this negative stain to show a capsule around a cell. The background stains, and there's the organism in there, but you see that little halo around it. That is a capsule. And having a capsule or not having a capsule can be an identifying feature of a microorganism. Bacterial flagella are a single thread of protein, and they are incredibly difficult to stain. But sometimes knowing the number and the arrangement of flagella on a cell is an important identification feature. In order to stain flagella, you first have to coat the flagella with metals and then stain that coating. This is a process that has many steps, it's very long, and it's only done if they really want to know how many flagella and how they're arranged on a cell. There are other ways we can determine if an organism has flagella or not that are much simpler. But here is a flagellar stain, and you see the many flagella. These happen to be paratrichously arranged because they're all around the organism. We've already talked quite a bit about fluorescent stains. We can develop an antibody to a particular cell or cell structure and then attach a fluorochrome to it. The fluorochrome will absorb the ultraviolet light and re-emit it at a visible wavelength that we can see. Because we can make antibodies to specific organisms, we can have not only a stain that helps us see things better, but also identify an organism. For example, we have fluorescent stains for Mycobacterium, and we have fluorescent stains for Legionella. Not only does it aid in the visualization, but we now know that just from staining that specimen, the suspect organism is in the specimen. Electron microscopes don't use dyes for staining, but they do use chemicals that contain atoms of heavy metals. These atoms will absorb electrons and alter how fast the electrons get through the specimen. Sometimes the chemicals bind to molecules in the specimen, sometimes they bind to the background. Some are very general, Others are specific for a particular molecule. These chemicals can also be attached to antibodies for more specific staining. Identification is the last step in processing our specimens. You start by looking at physical characteristics. Microscopic evaluations, the stains, will give you information about the shape of your cell and the composition of the cell wall. Your colonial characteristics will give you some idea about what you're dealing with. You can grow it on specialized media, you can look at the shape and the size of the colony. All of those can be features that help you identify your microorganism. Then you want to do biochemical tests. Now, this happens to be a test to see if the organism can ferment glucose, and the little upside down tube is to determine whether it can make gas from glucose. If it does, you get gas captured in the tube. No gas, and this one didn't ferment at all because fermentation gives you the yellow color. Biochemical tests can be done in large test tubes like this, or they can be miniaturized, and there are any number of commercially available systems for doing an array of biochemical tests on an organism so that you get enough information to identify an organism. Biochemical testing is still based on Berge's Manual of Determinative Bacteriology. It still goes back to that basis for identifying organisms. Sometimes you can do serological tests, and serological tests can be a couple of different types of tests. You can test the patient's serum to see if they have antibodies to a particular organism, or you can make antibodies to the organism and see if you can get the organism to react in some way. This is what we call an agglutination test, because while we couldn't see them here, once they agglutinate or clump up, we can tell that we had whatever our antibodies were against, that we have clumps of those. Phages are actually bacteriophages. Bacteriophages are viruses that infect bacteria. Viruses are usually very specific for the type of cell they infect, and some bacteriophages are so specific that they only infect particular organisms. If you have a lawn of bacteria, that is a smooth growth of bacteria, and you drop phages on it, as the viruses attack the bacteria and kill them, they leave these spaces where the bacteria have been killed. These are called plaques. So we can actually test bacteria against a variety of viruses and see which virus kills them. That can be another identification technique. Again, a technique not typically done in the clinical laboratory is analyzing the nucleic acids, actually looking at the DNA or the RNA. Initially, a very popular test was to check the guanine and cytosine content. That would help identify organisms, roughly putting them into one group or another. The final step of identification is actually figuring out what our organism is. That is, using some sort of taxonomic key. Most taxonomic keys are what we call dichotomous keys. These are simply a series of paired statements that you can answer yes or no to. So in this particular dichotomous key, we start out with the question, are they gram-positive cells? The answer here is no, so we ask about their shape. Are they rod-shaped? Yes. Can they tolerate oxygen? Yes. Can they ferment lactose? Yes. Can they use citrate as their sole carbon source? No. Do they produce gas from glucose? Yes. We have Escherichia. You can see that if we had come up with some different answers, we would have been taken to a different pathway. But this is one of the ways that we can use all of those biochemical tests to help us identify organisms.